afternoon, everyone. My name is Tuvik Beker. I'm the CEO of Pangea Biomed. By way of training, I have a PhD in machine learning, and I've spent the last 25 years applying AI technology to a variety of problems, focusing mainly on life sciences and healthcare. In Pangea, we try to solve one of the major issues in cancer treatment by developing better predictive treatment biomarkers. Now, as you know, precision oncology is perhaps the best hope for more than 50 million cancer patients living among us. And novel targeted drugs and checkpoint blockers can be very effective and sometimes bring complete response even at late metastatic stages of the disease, but the big picture is still grim. Using genomic matching based on a small number of actionable genomic events, current practices in precision oncology bring effective treatment to only about 15% of the patients. And the problem is not so much with the drugs, but with the way we match each patient with the most appropriate drug. Our vision in Pangea is a solution that can be applied very broadly to match most patients with more effective targeted and immune treatments. Enlight, the platform that we've developed over the last 5 years in close collaboration with the National Cancer Institute, does exactly that. Enlight offers the vast majority of patients treatment options selected from over 70 different targeted drugs and immune checkpoint blockers. And instead of focusing on individual gene abnormalities, Enlight matches treatments on the basis of deep analysis of gene expression patterns in the tumor. It has been broadly validated on numerous clinical trial cases in dozens of different cancer types, and as you can see at the bottom of the screen, these validations were published in some of the best peer-reviewed scientific journals. Pharma and biotech companies have been using Enlight to improve clinical programs by developing personalized response biomarkers to identify indications in which new drugs are likely to be more effective, subpopulations enriched with responders, and combination therapies with a synergistic effect. For example, here's a clinical trial with 3 different indications where an Enlight biomarker that was developed with no training on data from this novel asset correctly identified the patients more likely to respond to this bispecific antibody. Response rate in the overall population was 22%, but in the Enlight matched subpopulation, it was nearly 2 times higher at 38%, with a 77% sensitivity. Enlight NGS is our first clinical test, which was released last year after several years of testing in compassionate use settings. By analyzing RNA sequencing data from CLIA-certified NGS labs, Enlight NGS highlights treatment matches that may benefit patients who have exhausted the standard treatment guidelines. Enlight NGS provides physicians with a list of treatments matched individually to the patient's unique tumor, which should then be considered based on further evidence. The test has a guaranteed turnaround time of up to 5 days from the moment we receive the raw sequencing data, and it offers treatment matches in the vast majority of the analyzed cases. To assess just how often Enlight can offer matched treatments to advanced patients, we went back to the Winther trial of the WIN consortium that was led by Professor Kurzrock, where treatments were administered to 100 advanced cancer patients based on genomic and transcriptomic profiling. Enlight was able to accurately predict, based on the pre-treatment RNA data, which therapies would be effective, with nearly all the responders classified as Enlight matched and most of the non-responders classified as Enlight unmatched. In addition, Enlight was able to match 91 out of 100 patients with at least one therapy available in the trial, which in the case of the non-responders was almost always not the therapy chosen in the trial itself. But the evidence, this is kind of a counterfactual analysis, but by now evidence is not limited to retrospective data, because ENLIGHT matched treatments that contradicted standard biomarkers and guidelines have already saved the lives of multiple patients. And here is the story of Tal, who is one of the first among these.
למעשה, אמרו לי בבית חולים האחר שאני פשוט צריכה לחכות למות.
אני מציע לשלוח את הבדיקה של ה-RNA לחברה סטארט-אפ ישראלית, קוראים לה פנגאה. אחרי מספר ימים הם חוזרים עם תשובה שייתכן וטיפול אימונותרפי עם שתי תרופות יכול לסייע לטל.
הטיפול עבד והגרורות פחתו, ועכשיו אני במצב שאין בכלל זכר למחלה שהייתה. אומרים לך שאין לך סיכוי לחיות ואז יש חיים, אז הרגשה מדהימה.
And by the way, she was 36 when we analyzed the case. It's now more than 4.5 years later. She's leading normal life. She's free of the disease still. And ENLIGHT is able to to find treatment options where standard approaches fail because it examines the broader context in which drugs operate. Instead of focusing only on the direct targets of the drug, the ENLIGHT genetic interactions engine scans terabytes of cancer data to reveal interactions between some 200 million gene pairs, interactions that affect tumor fitness. And it then uses these insights to find the tumor expression state that best suits each drug. By comparing the expression in a given tumor to that desired network state, ENLIGHT can assign each drug a personalized tumor matching score. In a work that was recently accepted for publication, we systematically investigated ENLIGHT's ability to broaden the scope of precision oncology biomarkers. Out of 114 drugs that have clearly characterized genetic targets, only 7, or about 6%, had coupled transcriptomic and response data that could be the basis for training supervised learning models. In contrast, ENLIGHT biomarkers could be developed for all of these drugs and were proven predictive in 95% of the cases that were tested. For the 7 cases where both approaches could be used, the performance of the ENLIGHT unsupervised markers was on par with the supervised approach. In the last couple of years, we've been working on a new technology which replaces RNA sequencing with digital pathology. This deep learning architecture called DeepPT takes high-resolution scans of standard H&E slides and translates their morphological features into a detailed profile of gene expression in the tumor. The resulting test, called ENLIGHT-DP, is similar to ENLIGHT-NGS, but it has the advantage of using much more accessible and easy to obtain input data. In a paper that we published last year in Nature Cancer, we demonstrated the ability of ENLIGHT-DP to predict response to a variety of different drugs in 5 cancer types, based on a single H&E slide from a biopsy taken weeks to months before treatment started. ENLIGHT-matched therapies had 2.8 times higher odds of response than their unmatched counterparts. A few weeks ago, we published another validation of ENLIGHT-DP, this time in a clinical trial of 50 metastatic non-small cell lung cancer patients who were treated with first-line anti-PD-1 plus chemotherapy. The ENLIGHT matching score proved vastly superior to standard biomarkers like PD-L1 expression measured by immunohistochemistry or tumor mutational burden measured by next-generation sequencing. Not that it's such a high bar to surpass. But moreover, ENLIGHT was particularly good at identifying responders in subpopulations where the standard biomarkers were negative. So we can find those rare responders. The superiority of the ENLIGHT matching score was not limited to objective response, but it manifested itself also in the patient's overall survival and time to progression. Whereas you can see that the standard biomarkers completely failed to achieve any meaningful patient stratification for these two important outcome measures. In a clinical trial that's currently ongoing in Sheba Medical Center, we are testing the ability of ENLIGHT-DP to guide functional assays. What happens in the trial is that slides from resections or biopsies are analyzed by ENLIGHT-DP within 24 hours of sample collection. And the list, the prioritized list of matched treatments that we produce is added to the list of drugs requested for testing by the physicians. Out of 26 cases analyzed so far, in more than 15, ENLIGHT-DP uniquely suggested targeted treatments that were corroborated in the 3D functional assay. And we have so far outcome data for 6 patients, and out of these, in 4 cases, ENLIGHT-DP correctly predicted the outcome, 2 non-responders and 2 responders. Now, this trial was not planned as an interventional one, but in one of the cases, there was no sensitive drug in the functional assay except for a drug brought up by ENLIGHT. And the physician said, I've never prescribed this drug. It was a mucosal melanoma case. But since this patient already resisted 3 lines of treatment and that's the only option, let's try it. And the patient has already been in remission for 9 months. One of the exciting advantages of our digital pathology analysis is that it allows us to examine spatial patterns of expression and how they affect the ENLIGHT matching score. We've already seen that the approach can reliably reproduce the spatial expression patterns of thousands of genes, and that heterogeneity in gene expression has a significant predictive power. Now, together with our collaborators at the NCI, we're investigating how this novel application can further improve the quality of our predictions. In 2025, we are planning to significantly expand the distribution of our ENLIGHT NGS tests, beginning to sell tests in the UK next month and in the European Union, India, and the US later this year. We are working on setting up reference CLIA certified labs in several countries, which will stain and scan histopathology slides to support the distribution of ENLIGHT-DP tests. And the NCI recently received FDA clearance to go forward with the synthesis trial, which is an interventional trial of ENLIGHT. And we hope to launch the trial next quarter. Finally, we are hoping to see the first IND trial that will use an ENLIGHT-based companion diagnostic test as part of its design. I want to mention the wonderful group of renowned scientific advisors who are helping us develop, refine, and validate ENLIGHT. It is a great privilege to be working with such a team of clinicians and cancer researchers, and special thanks to Professor Kurzrock, who's always been very generous with her time and encyclopedic knowledge, and contributed to some of our key publications. Finally, I want to thank the amazing team at Pangea who's made all this possible, and the many others who have collaborated with us on works featured in this presentation, and thank you all for listening.